Good morning, I am Madhurima Datta, a zoology and biology educational content creator. Browse content for B.SC Zoology Honours, M.SC Zoology Honours, CVT PG and CSI NET Life Sciences students. Paper Non-Codex, Unit Mollusca and topic is class Bivalvia. Introduction. Bivalvia are also known as Pelecypoda or hatch-foot animals as their name implies. Their size, they are bivalve mollusks that include mussels, clams, scallops, oyster, and shipworms, and they range in size from teeny seed shells 1 to 2 millimeter in length to giant South Pacific clams. Habitat. Most bivalves are marine, but many live in brackish water and in streams, ponds, and lakes. Form and function of bivalves. Shell, the outer body structure of the bivalves. Bivalves are laterally compressed and their two shells, that is called valves, are held together dorsally by a hinge ligament that causes the valves to gap ventrally. The valves are drawn together by adductor muscles that work in opposition to the hinge ligament. The umbo is the oldest part of the shell and growth occurs in concentric lines around it. Next, important mechanism for B.SC Zoology Honours and M.SC Zoology, that is the pearl formation. First point is the intrusion. An irritant, most commonly a teeny microscopic organism or parasite, penetrates the mollusk shells and enters the soft tissue, usually the mantle. The mollusk immune system recognizes this intruder as a threat. To protect its delicate tissue, it isolates the foreign body within a specialized tissue sac known as pearl sac. Next point, secreting nacre. The mantle tissue lining the pearl sac begin to secrete nacre, also known as the mother of pearl, over the irritant. Nacre is the biomineral made of microscopic aragonite crystal glued together by an organic protein matrix called conchiolin. Next point is layering. Layer by layer, the mollusk continuously deposits nacre around the irritant over several months to a few years. Thousands of these thin reflective layers build up and resulting in a smooth iridescent pearl. Next point, body and mantle of bivalves. The visceral mass is suspended from the dorsal midline and muscular foot is attached to the visceral mass anteroventrally. The tenidia hang down on each side, each covered by a fold of the mantle. The posterior edges of the mantle folds are modified to form dorsal excurrent and ventral incurrent opening. Locomotion. Bivalves initiate movement by extending a slender muscular foot between the valves. They pump blood into their foot, causing it to swell and to act as anchor in the mud or sand. Next point is respiratory system. Gaseous exchange occurs through both mantle and gills. Gills of most bivalves are highly modified for filter feeding. They are derived from primitive ctenidia by a great lengthening of filaments on each side of the central axis. As ends of long filaments became folded back toward the central axis, ctenidial filaments took the shape of a long slender W. Next, filaments lying beside each other became joined by ciliary junction or tissue fusions, forming a plate-like lamella with many ventral water tubes inside. Thus, water enters the incurrent siphon propelled by actions, then enters the water tubes through the pores between the filaments in the lamella, proceeds dorsally into a common suprabranchial chamber, and then out the excurrent aperture. Next point, feeding of the bivalves. Most bivalves are filter feeders. Respiratory current bring both oxygen and organic materials to the gills, where ciliary tract direct current to the tiny pores of the gills. Gland cells on the gills and labial pulp secrete copious amount of mucus. which entangles particles suspended in water going through gill pores. This amount masses slide down the outside of the gills toward the food groups at the lower edge of the gills. Heavier particles of the sediment drop off the gills as a result of gravitational pull, but small particles travel along the food groups toward the labial pulps. The pulps, being also grouped and ciliated, sort the particles and direct tasty ones encased in the mucous mass into the mouth. Next, internal structure and function. The floor of the stomach of filter-feeding bivalves is folded into ciliary tracts for sorting a continuous stream of particles. In most bivalves, a cylindrical style sac opening into the stomach secretes a gelatinous rod called a crystalline style. This is an important question. What is the crystalline style of bivalves? Which projects into the stomach and is kept whirling by means of the cilia in the style sac. Next, their mechanism is rotation of the style helps to dissolve its surface layer, freeing digestive enzymes that it contains, and to roll the mucous food mass, and dislodged particles are sorted and suitable ones are directed to the digestive gland or engulfed by amoebocytes. Further digestion is intracellular. Next, circulatory system. The three-chambered heart, which lies in the peripheral cavity, has two atria and ventricle and beats slowly, ranging from 0 to 2 to 30 times per minute. Next, excretory system. A pair of U-shaped kidney, this is called nephridial tubules, lies just ventral and posterior to the heart. The glandular portion of each tubules opens into the pericardium. The bladder portion empties into the suprabronchial chamber. Next, for nervous system, the nervous system consists of three pairs of widely separated ganglia connected by commissures and a system of nerves. Sense organs are poorly developed. Now the topic has ended. For the all videos of B.SC Zoology Honors and M.SC Zoology, please subscribe my YouTube channel Biogenesis by Madhurima. I share the link in the comment box of my YouTube channel and follow my Facebook profile Madhurima.